Very good evening to one and all. Welcome to the POPMA Study Club, date 27th February 2025 from 4 to 5 p.m. And the study club is organized under the aegis of POPMA, and POPMA is Practicing Oral Pathologists and Microbiologists Association. Those who are new to the channel, I am Dr. Bhuvan Nagpal, and I am the founder president of POPMA and the chairperson for Oral Pathology Advocacy Committee, which includes POPMA Study Club. We have our co-chairperson, Dr. Ketki, and moderator for the session, Dr. Nidhi Jadhav ma'am. So to introduce the author, the speaker, because Dr. Nidhi ma'am was a bit stuck, and so I am gonna introduce Dr. Sharon. Dr. Sharon is the guest speaker, and she is an enthusiastic oral pathologist, a cancer biologist, educator, and researcher with expertise in head and neck pathology, molecular oncology, and clinical diagnostics. She is currently pursuing PhD in cancer biology at the University of Alabama at Birmingham, and she has completed MDS in oral pathology with second university rank, and she also holds a fellowship in head and neck pathology from Malabar Cancer Center, Kerala. She has extensive experience in histopathology, cytology, immunohistochemistry, and molecular diagnostics. Her professional journey includes a role as a fellow at Malabar Cancer Center, senior program manager at Mazumdar Shah Medical Foundation, and also lecturer at the University of Technology, Jamaica. Dr. Sherin is an award-winning researcher with multiple international publications, and she has been actively involved in clinical trials, translational research, curriculum development, and scientific writing. She's a she's a Sparkman Fellow at University at Alabama at Birmingham, and a recipient of ICMR Research Award. Over to you, Dr. Sherin, and please go ahead.
Thank you, Dr. Bhuvan, for the introduction. It's truly a pleasure to be here. You can share your screen. Sure.
Dr. Sherin?
Yes, sir. Are you able to hear me? And I've shared the screen right now.
Yes. I request everyone to please put your cameras on off mode. Yeah, over to you, Dr. Sharon. You can go ahead.
Sure, sir. Once again, thank you, Dr. Bhuvan, for facilitating this. And I'll be talking about the biology of cancer, mechanisms, evolution, and emerging insights. I've organized today's session into 4 distinct segments, each addressing a clinical aspect of cancer biology. First, we will explore the biological revolution and its impact on cancer research. Then we'll delve into what makes cancer unique, a closer look at the characteristics that set cancer apart from other diseases. And following that, we will examine some of the emerging frontiers in cancer research, highlighting some later developments and cutting-edge technologies. And finally, I will share my insights on why pursuing a PhD in cancer biology is not only a personal journey, but also a vital contribution to the advancing field. So the biology of cancer research, or the biological revolution of the 20th century, totally reshaped all fields of biomedical study, and cancer research is definitely one of them. And this revolution, which began in the mid-century, was triggered by Watson and Crick's discovery of DNA double helix. Today, our understanding of how cancer arises is continuously being evolved by the discoveries in diverse fields of biological research. In a nutshell, cancer cells are normal cells that have gone rogue, and they grow uncontrollably to form a tumor. So what a normal cell tries to do is to carefully program to collaborate with one another in making a diverse tissue whereas cancer cells have just one agenda, that is to make more copies of themselves. So I would like to quote Dr. Weinberg here. He calls cancer as a disease of chaos. It is important to know that the field of tumor phylogenetics has evolved considerably since Peter Nowell first introduced the clonal evolution model in 1976. This model suggests that tumors originate from a single abnormal cell. In general, tumor evolution is a process that is driven by both genetic and epigenetic mutation that results in a complex of evolving mosaic of cell population that we call as subclones. Each subclone has a unique genetic and phenotypic profile, and this contributes to the tumor's overall heterogeneity, which leads to the genetic diversity. So, according to the phylogenetic tree of cancer evolution, so here you can see the gray circle in which it represents a normal cell, and the central dot depicts the initiating somatic mutation that drives the founder clone. So the different colored circles you see represent subclones that have accumulated successive mutation. It is good to note that here in the primary tumor, there are several subclones that coexist. Although some expand, notice that there are some other that remain dormant or become extinct. And metastasis can originate from either a major clone in the primary tumor, that is metastasis 1, or from a minor clone, that is metastasis 2. You can also see that even in the metastasis stage, there is a clonal evolution that's happening. We know that the population of cells that are derived from a common ancestral cell is called as monoclonal. And the second image here, it shows that in a monoclonal tumor, not only a single cell is transformed to become the ancestor of this other cells in the tumor mass, and some of the classic examples for this theory are myeloid cancers like CML and multiple myeloma, etc. Whereas in a polyclonal tumor, multiple cells cross over the border from normalcy to malignancy, and they become several genetically distinct subpopulations that constitute a tumor mass. Concepts like field cancerization and the interactions between epithelial, stromal, and immune cells contribute to this multiclonality. So it is via tumor heterogeneity that is observed through single cell sequencing and lineage tracing that the polyclonal hypothesis was supported. Most of the solid tumors that we know are examples of this theory. So to give a perspective, we have more than 37 trillion cells in our adult body. All of them carries genetic information. And so logically, this explains why there is always a risk for disaster to strike. In that case, it would seem logical that the human population throughout the world would experience similar frequencies of cancer. But the cancer incidence rates in various countries seem to vary dramatically, and we need to be cautious here just simply because incidence rates of certain cancers may be highly misleading. And this is a consequence of diagnostic bias. I can explain with an example here. So, for countries like Australia that have some of the highest cancer incidence rates, often due to lifestyle factors, aging populations, and most importantly, they have efficient cancer registry and robust screening programs. While in developing countries, underdiagnosis and underreporting could show a low incidence. The process of tumor formation happens in multiple steps. Citing an example of colon cancer here, you see normal colonic epithelium, adenoma, and carcinoma stage. And one of the several driver genes that encodes between the transitions or of that denotes a major signaling pathway are shown here, and the patient age is indicated to show the time intervals during which the driver genes are usually mutated. Similarly, In the second model, there is an ordered histological progression of head and neck SAC. There is mucosal epithelial hyperplasia, followed by dysplasia, and carcinoma in situ, which precedes carcinoma. There are specific genetic events that are found to be enriched at each stage of transition or progression. And it's interesting to see that, unlike in most cancer, in head and neck cancer, its oncogenic mutations are not what typically drives tumorigenesis. It is usually the inactivation of tumor suppressor genes like cyclin dependent kinase 2A, TP53 in early stages, and in late stages, phosphatase tensin homolog inactivation are some of the causative or driver mutation in the major signaling pathway. So catching up with a little bit of history here, English surgeon Dr. Percival Pott's work in 1775 proved that coal tar condensates were implicated for scrotal cancer. And this was a causation in chimney sweepers during those days. And this inference was used by a Japanese pathologist, Kazusaburo Yamigawa, in 1915, where he repeatedly painted a rabbit's ear skin with chemicals to induce skin cancer. So the image here shows Dr. Yamigawa's world's first artificially initiated cancer on a rabbit's ear, which is preserved at the University Museum of Tokyo. Unfortunately, he was little noticed in the international scientific community of that time, and the 1926 Nobel Prize in Medicine was given to a Dane whose research on experimental induction of cancer was soon discredited. In retrospect, Yamigawa's work, together with Peyton Rose, about whom we will discuss in the coming slides, they demonstrated that cancer could be induced at will in lab animals. The interesting stuff or the take-home message from the slide is that before all these breakthroughs, researchers had to wait passively for months together for tumor to appear spontaneously in wild or domesticated animals to study them. And this is so amazing because at present, we have various human and mouse cancer cell lines along with xenograft, syngeneic, humanized mouse models to have a controlled tumor induction in vivo that significantly accelerates our cancer research. While major discoveries were being made, there was an independent line of research that suggested that cancer might be an infectious disease. So as early as 12th century, researchers observed that viruses could cause leukemia and sarcoma in chicken. So this led to three competing theories at that time on the origins of human cancer. One is viral, chemical, and radioactive causes. The confusion surrounding carcinogenesis theories was significantly reduced in 1927 when Dr. Hermann Muller started working on fruit flies, and he demonstrated that an animal's genome could undergo mutation. And this discovery established that genetic changes could play a role in disease development. By late 1910s, during the time of World War I, from 1914 to 18 specifically, mustard gas was used as a chemical warfare. And many of these gases were alkylating agents later found to be mutagenic. And further research confirmed that carcinogenic agents like PAH, polycyclic aromatic hydrocarbons, aromatic amines, and azo-dyes, they could induce cancer by mutating DNA. This led geneticists to speculate that cancer was a disease of mutant genes. Later, Hermann Muller also demonstrated that even radiation can cause DNA damage. But a major breakthrough came with the discovery of abnormal chromosomes in CML, and this abnormality was soon called the Philadelphia chromosome, which was named after the place it was discovered at Fox Chase Cancer Center, Philadelphia, by Dr. David Hungerford. And this finding actually provided a definitive proof that cancer is fundamentally a disease of genetic mutations. Coming back to the infectious origins of cancer, in the late 1910s, Peyton Rose at the Rockefeller Institute, he made a discovery. He identified that raw sarcoma virus, an RNA virus, caused the formation of cancer-like foci in cultured normal chicken cells. And this was for the first time that cancer could be studied at the level of the cell. And fast forward to 1970s. This is when Michael Bishop and Harold Warmers, they analyzed RSV and discovered that its oncogenic potential was due to an extra gene called viral SRC or VSRC. So their key finding was that this viral SRC was not entirely viral in origin, but this was actually derived from a normal cellular gene that was present in chickens, which they named as cellular SRC or CSRC. And this was a pivotal moment in cancer biology because this established that normal cells contains proto-oncogenes, which you and me have, and this undergo specific mutation, which could be converted into an oncogene that drives uncontrollable cell proliferation. So this discovery laid the foundation of all of the modern oncogene research and demonstrated that cancer can arise from mutations in normal cellular genes. So for their groundbreaking work on cellular origins of oncogenes, Bishop and Warmers were awarded the Nobel Prize in Physiology in 1989. And later, it's Dr. Robert Weinberg who further advanced this field by identifying the first human oncogene, RAS, and the first tumor suppressor gene, retinoblastoma. And our current understanding of cancer is revolutionized by their findings, which has paved the way for targeted therapies that focus on oncogene-driven malignancies. So now that we know that tumor viruses induces cancer, but how do these multiple changes occur in a cell phenotype? So the image here shows a normal chicken embryo fibroblast in blue that are growing on the bottom of a petri dish. And they form a one cell thick monolayer because these cells stop proliferating when they touch each other. And it's a behavior that we know as contact inhibition or topo density inhibition. But if one of these cells were infected by RSV before reaching confluence, the cell in red color, and its descendants acquire rounded morphology and they lost contact inhibition. So as a consequence, they continue to proliferate and eventually accumulate in a multi-layered clump of cells that are even visible to the naked eye. So the effects of RSV infection could be observed through phase contrast microscopy, where we see normal chicken embryo fibroblasts that are spread out and form a continuous monolayer. And in scanning electron microscopy, they round up and become refractile and pile up on one another. So like RSV transformed chicken cells, the rodent cells transformed by simian virus 40, they showed a rounded shape, a loss of ability to grow over one another, ability to grow without an attachment to solid substrate, ability to proliferate indefinitely, reduced requirement for exposure to mitogenic growth factors, high saturation density that allows to accumulate large number of cells in a culture dish, inability to halt proliferation in response to deprivation of growth factors, increased import of glucose, glycolysis in the presence of oxygen, and tumorigenicity, that is an acid test for cellular transformation. So as we explored here, these distinctive characteristics of transformed cell in a cell culture, you might recognize a familiar pattern. So these were the defining traits that were conceptualized by Doctors Hanahan and Weinberg into framing the famous hallmarks of cancer, and by drawing inspiration from the very observations from the many results of experiments we are discussing today. So let's move on to part 2 on what makes cancer exclusive. Before that, I thought of doing a trivia question for y'all. So can anybody in the audience recognize this image, either the people or what this image was about? You can either put in the chat box or maybe unmute and answer it. All
I
right.
think Dr. Sherin, nobody has answered.
No,
Anybody
sir.
wants to say, you can go ahead with that. Either unmute or you can put it in the chat box.
Hello.
Yes, Dr. Kiran. Yes,
I
sir.
don't know the exact name of the person, but is it related to the diabetes?
Oh, excellent. Excellent, sir. That's fantastic. Yes, this is
Insulin.
banding and best. Yes.
Yes, yes,
Oh,
insulin.
Yes, with their
yeah.
that's fantastic. Yes, and this was the first dog that was ever treated with insulin in 1921.
Yes,
And in a
yes.
year, yeah, they treated the first child, a 14-year-old
Yes.
child, and that was revolutionary. Yes, yeah,
Mm.
that's fantastic, sir. Yeah.
Even one more answer has come from the chat, but the name is not AMW. It is written Banting.
Done.
Yeah.
Oh, yes, that's Dr. Banding. Actually, this is Best Charles Best was his medical student at that time. So he couldn't win Nobel Prize when that was given, but Dr. Banding was kind enough that he shared his Nobel Prize money with his medical student later, which I found interesting. So yeah, coming back to what we were discussing on. Um, so one of the most important papers or foundation paper for um, MDS students or students who are interested in cancer research is to understand the code hallmarks of cancer, which came out in 2000, which was revolutionary at that time. I'm not going in detail on these hallmarks because we already had a fantastic presentation on this POPMA Study Club in 2024, where Dr. Yoidab explained each of the hallmarks with excellent illustrations. So later, after 11 years, Dr. Hanahan and Weinberg again modified the hallmarks and called it as the next generation. And notice that they put forward two emerging hallmarks and two enabling characteristics. So what is the difference between them? So emerging hallmarks are the newly recognized biological capabilities that cancer cells acquire as the disease progresses. And the capabilities by which these hallmarks are acquired are called as enabling characteristics. So they are basically supporting factors that facilitates the acquisition of the core hallmarks, or they provide a permissive environment for the cancer to progress. And this is a hallmark flower, which at that point included all the 10 hallmarks of cancer. So again, 11 more years later, Dr. Hanahan alone came up with the new dimension, which included another two more emerging hallmark and two enabling characteristics. And these are the combined 14 hallmarks of cancer that we have now, 6 plus 4 plus 4. Please remember these hallmarks thoroughly. But what makes cancer exclusive? So, despite overlapping mechanism, cancer is the only disease that evolves and adapts, which means cancer is not static. It mutates, it evolves, and it develops resistance like an evolving species within the body. It has unlimited replicative potential where telomere shortening limits the lifespan of
Moitzer
normal,
later, Saman.
including those in neurodegeneration. But cancer cells can reactivate telomerase, allowing infinite divisions, making them immortal. They have metastasis ability, which which shows where cells detach and survive in circulation and colonize in distant organs. And they are resistant to death. Cancer can actively block apoptosis either via p53 mutations or by the overexpression of anti-apoptotic proteins like BCL2. And they can hijack the microenvironment where they reprogram the surrounding tissue for its benefit. So, in conclusion, cancer is a disease of evolutionary advantage. There was a 2023 article on hallmarks of cancer, a New Testament from the Queen Mary University of London that has included many of the 2022 new dimensions hallmarks like epigenetic dysregulation, altered microbiome, and altered phenotypic plasticity, which was added with our 2022 hallmarks modified. But one additional point that they added that was not governed in the original hallmarks is that neurons and their axonal projections could be enabling constituents of tumor microenvironment. They showed that tumors are also able to induce neural remodeling that encourages nerve ingrowth and increases neuronal excitability and therefore reinforcing nerve cancer interaction. The perineural invasion that we see in histopathology has established that innervation of the tumor microenvironment alongside the neuronal signaling in cancer can modulate six of the current core hallmarks parameters and could be an exciting frontier in cancer biology. And hopefully in 2033, in another 8 more years, we can expect another seminal article with more hallmarks added together. And I said 2033 is because I see a pattern of 11 years, a gap of a decade when when they keep advancing, because uh, there is a need for added evidence. It generally takes a decade for them to update, I guess. So here I have categorized cancer hallmarks based on their role in initiating, sustaining, and defining cancer as a disease. This approach just reorganizes hallmarks based on their functional role in cancer progression, but note that most of the hallmarks actually overlap between these stages. It's not a hard and fast rule that, okay, this specific hallmarks only initiate or drive the process. No, it can overlap. But the point I'm adding this was for students to have an easier way of remembering these hallmarks when asked in exams. So hallmarks that drive or starts a process, we can consider sustained proliferative signaling, which allows cancer cells to grow uncontrollably, evading growth suppressors that removes the brakes on cell division, genome instability and mutation that drives cancer by accelerating the acquisition of tumor promoting mutation and letting it accumulate unchecked, tumor promoting inflammation where inflammatory cells releases reactive oxygen species which induces DNA damage and accelerate genetic mutations causing increasing instability and promoting malignant evolution, non-mutational epigenetic reprogramming with hypoxia, nutrient deprivation, and even extracellular metric stiffness that can induce broad epigenetic changes like DNA methylation or chromatin remodeling. All these enhances the tumor fitness and they initiate the malignant transformation by reprogramming their cell. And polymorphic microbes like H. pylori in gastric cancer and Fusobacterium nucleatum in colorectal cancer are examples to show how they contribute to carcinogenesis by inducing either inflammation, DNA damage, or immune evasion. Like I said, many of these hallmarks do overlap. For example, if you consider that gut dysbiosis, they can promote chronic inflammation and support tumor progression too. So it is not just fitting into one category. So the hallmarks that maintains cancer, which allows the growth and spread and sustenance of the tumor by ensuring its continuous growth and expansion, are resisting cell death, where it prevents apoptosis and allows damaged cells to survive, enabling replicative immortality that prevents cells from aging and dying, inducing angiogenesis, which provides oxygen and nutrients for the cancer cells to grow, deregulating cellular metabolism that allows cancer to survive in harsher environments, avoiding immune destruction that helps tumors evade immune system attacks, and senescence associated secretory phenotype that releases cytokines, chemokines, and growth factor, which promotes inflammation, immune suppression, and all the factors that aids in tumor progression. And what are the hallmarks that distinguish cancer from normal cells or that defines the unique characteristics? One is definitely activating invasion and metastasis that separate it from benign tumors and unlocking phenotypic plasticity. For example, the transdifferentiation that happens in Barrett's esophagus, where chronic inflammation triggers the squamous epithelial esophageal cells to convert or transdifferentiate into an intestinal-like columnar epithelium. And this creates an environment for adenocarcinoma instead of a squamous cell carcinoma to be formed. And this distinguishes cancer by shifting cell lineages and predisposing it into a different tumor type than expected. Another interesting way to look is that cancer cells often simulate developmental pathways seen in embryogenesis. And this includes cell polarity, lineage commitment, stemness, plasticity, and all of this enable them to evade differentiation and sustain uncontrolled growth. So tumor progression mirrors early developmental programs such as epithelial mesenchymal transition and chemotherapy hydration, which are crucial for both embryonic tissue organization and cancer metastasis. Moreover, there are multiple signaling pathways like Wnt, hedgehog, notch, all of which regulate stemness and differentiation during development and are frequently reactivated in cancers, driving tumor heterogeneity and therapy resistance. So the gist from the slide is that cancer hijacks our embryonic developmental pathways. So one of the most insightful tumor classification system I have come across is this one, which categorizes cancer based on differentiation state, embryonic origin, and biological activity. So based on differentiation state, they distinguish tumors as epithelial, mesenchymal, and mixed, and embryonic origin into ectodermal, mesodermal, and endodermal, while biological activity considers growth kinetics and invasiveness and metastatic potential, which is a cool way for beginners to understand the hierarchy of cancer. And the intracellular circuitry in cancer cells, they operate as nodes and branches of integrated circuits. That is a sentence exactly from our Hallmarks of Cancer paper. So any signaling pathway, it is important to know, can be broadly viewed in 3 steps. So the first is signal reception, where an extracellular ligand binds to the receptor on the cell membrane. Signal transduction, that is when the ligand and receptor get attached, they get activated and trigger a cascade of events or signaling events that involves phosphorylation, etc. And downstream molecules are being activated. And secondary messengers are important here because these are small molecules like calcium ions or inositol triphosphate that can amplify the signal and transmit it throughout the cytoplasm. And finally, cellular response, where you see a specific response like changes in gene expression, cell growth, or differentiation. So one of the key signaling pathways in head and neck cancer involves epidermal growth factor receptor, which is a protein located on the cell membrane and functions as a receptor tyrosine kinase, RTK. So the ligands for the EGFR pathway are EGF, TGF alpha, transforming growth factor alpha, amphiregulin, beta-cellulin, etc., which binds to EGFR. And this results in the receptor dimerization. It can be either homo or heterodimerization, depending on what kind of receptors attach, because the ERB family has 4 subsequent ones like HER1, HER2, HER3, and HER4. HER2, which we know is important in breast cancer. So EGFR is and another name for HER1. So after this receptor dimerization occurring, there is autophosphorylation of tyrosine residues in the cytoplasmic region. So whenever you hear of autophosphorylation, just remember that it's a process where a kinase adds a phosphate group to its own amino acid residues. And it's mostly either tyrosine, serine, or threonine. And this happens without the involvement of another kinase, and that's why it's called as autophosphorylation. And this provides docking sites for signal transducers such as RAS to bind to the signaling cascade and induce gene transcription. The downstream signaling cascade of EGFR can be broadly divided as the RAS-RAF-MEK-ERK pathway, or the PI3K-AKT pathway, or the JAK-STAT pathway. And there are a lot of targeted therapies against EGFR that was developed. And for example, is cetuximab or nimatuzumab, which are monoclonal antibodies that targets the extracellular domain of EGFR. Thereby, it physically blocks the EGF or the growth factor to bind on the receptor, whereby it inhibits tumor cell proliferation and survival. So cetuximab is generally used in metastatic colorectal cancer and even in head and neck cancer. And there are EGFR tyrosine kinase inhibitors like afatinib and gefitinib, where they bind to the tyrosine kinase domain of EGFR on the intracellular portion of the protein. So here comes the side villain of the show, I would say, that is tumor microenvironment. So the global sequencing efforts that were done initially, such as the International Cancer Genome Consortium, they improved our knowledge of specific mutations in tumor cells. So initially, the reductionist view was to just focus on the cancer cell. But what we are actually dealing with is a complex heterotypic tumor microenvironment where tumor cells are just the foundation of cancer as a disease, but not its complete manifestation. So cancer, as we know today, is the aggressive and therapy-resistant one. And that is because it is constituted of different kind of cells, which includes cancer stem cells, which are special subclass with enhanced tumor seeding ability and phenotypic plasticity. And this offers resistance to chemotherapy and contribute to tumor dormancy and recurrence. And there are endothelial cells that are regulated by a VEGF, FGF, angiopoietin, etc. They show poor functionality like leakiness, including lymphatic vessels, which is usually collapsed within tumors. Then pericytes that wrap around the blood vessels and provide support to the endothelium to maintain vascular integrity. And immune inflammatory cells, the tumor promoting cells, especially M2 macrophages, neutrophils, mast cells, and not to forget partially differentiated myeloid progenitors, which are bad guys. Then cancer-associated fibroblasts, which includes our fibroblast and myofibroblast that undergoes modification and supports invasion and metastasis and contributes to desmoplastic stroma formation. Then there are stromal stem and progenitor cells that can either differentiate from adjacent normal tissue or bone marrow, and they can differentiate into various stromal cell types while maintaining the undifferentiated states with unique function. And lastly, blood vessels, lymphatics, extracellular matrix, nerves, microbiome, all this is what actually constitutes the tumor. So it's fascinating that some cells in tumor microenvironment, they exhibit dual roles. They act both as tumor suppressor and tumor promoter. In short, they act as a double-edged sword. And one of the famous characters here is TGF-beta, or transforming growth factor beta. In earlier stages, they have a tumor suppressor role where they inhibit proliferation by inducing cell cycle arrest and promoting apoptosis through SMAD-dependent pathways. They also enhance DNA repair mechanisms. But in later stages, TGF-beta takes up a tumor promoter role. It induces EMT, epithelial mesenchymal transition, and they also suppress immune surveillance by promoting regulatory T cells. Again, another bad guy in terms of cancer. Another component is autophagy that plays a paradoxical role in cancer. It brings up a question of survival or death mechanism. So autophagy, certain studies show, has a tumor suppressor role where they clear the damaged organelles and misfolded proteins, and they prevent the genomic instability from happening. And they can also eliminate some precancerous cells via autophagic cell death. And as a tumor promoter, they key regulators like Beclin-1, LC3, they provide metabolic adaptation during nutrient starvation and hypoxia, and they help cancer cells to resist chemotherapy by removing the toxic cellular components. Necrosis was considered once as an unregulated form of cell death, but recent studies show it's more than just that. What we have studied is that anti-tumor necrosis, there is necrotic death of tumor cells, and this in turn can trigger pro-inflammatory responses that recruits CD8 T cells and induce immunogenic cell death, and thereby it enhances the immunity against cancer. But the pro-tumor necrosis is in such a way that necrotic tumor cells release damage-associated molecular patterns that activates inflammatory response, and they also recruit myeloid-derived suppressor cells that contributes to immune evasion. Next is macrophage. I like to call macrophage as the yin-yang of cancer because they are among the well-studied immune cells in cancer, and their polarization is influenced by tumor microenvironment cues, hypoxia, and tumor-secreted cytokines. So the M1 macrophage has an anti-tumor role where they secrete pro-inflammatory cytokines like TNF-alpha, interferon gamma, and they promote cytotoxicity. And they also induce tumor cell apoptosis, whereas the M2 macrophages play an important role in pro-tumor. They secrete interleukin 10, TGF-beta, and they suppress the anti-tumor immunity. And they can also promote angiogenesis via VEGF and matrix metalloproteinase 9 secretion. And another factor is neutrophils, which N1 neutrophils are anti-tumor. They produce reactive oxygen species and enhance cytotoxic T cell recruitment. And we have the N2 or the tumor-associated neutrophils, just similar to tumor-associated macrophages. And they promote angiogenesis and metastasis via VEGF and interleukin 8 secretion by suppressing the CD8 T cell function. Fibroblast or cancer-associated fibroblast. The normal fibroblasts, as we know, they maintain the tissue integrity and suppress proliferation, whereas CAF cells, they secrete TGF-beta or heparin growth factors, and they enhance the cancer stemness and metastasis and promote the immune evasion by recruiting T regulatory cells. The last kind of cells, the T cells, are of different types. And we know that effector CD8 T cells or cytotoxic T cells are good. They kill the tumor cells via perforin and granzyme secretion, whereas regulatory T cells are pro-tumorigenic, where they suppress the immune activation via interleukin 10, TGF-beta, and allows the tumor to evade immune destruction. So angiogenesis, another component in tumor microenvironment, involves a complex interplay between various cell types that is signaling to each other and responding like a dialogue rather than a single cell type that solely drives the process like a monologue. So few names that I would like y'all to remember when it comes to the field of angiogenesis is that of Dr. Judah Folkman, who is known as the father of angiogenesis, since he proposed in 1971 that cancer rely on new blood vessel formation for growth and metastasis, and his groundbreaking work led to the foundation of anti-angiogenic therapies. Next is Dr. Napoleon Ferrari and his team at GeneTech. They first isolated and cloned VEGF in 1989. And in 1993, they showed that blocking VEGF with monoclonal antibodies significantly suppressed the tumor growth. And this led to the development of a humanized anti-VEGF antibody, Avastin, in 1997. And today, Avastin, also called as bevacizumab, is widely used to treat different solid tumors, and it has revolutionized care for millions of patients with age-related macular degeneration worldwide. Another giant in this field is Dr. Rakesh Jain, who in 2005 introduced the vascular normalization theory, showing that anti-angiogenic therapy can actually restore the normal blood vessels transiently to a more functional state. And this would improve the drug delivery and immune response. And this research has reshaped cancer therapy by optimizing the efficacy of both chemo and immunotherapy. So the image here shows the proposed role of vessel normalization by Dr. Jain in response to anti-angiogenic therapy. In subsection A, you see tumor vasculation, a normal one, and an abnormal one, which is functionally abnormal. However, a sustained or aggressive anti-angiogenic regimen eventually prune away these vessels, where they initially improve both the structure or function of the cells here in the normalized. In the subsection B, you see the dynamics of vascular normalization that is induced by VEGF blockade. And this is normal vessels. These images are two photon image that is showing normal blood vessels in a skeletal muscle. And the subsequent image shows a human colon cancer vasculature in mice at day 0, day 3 after giving the VEGF treatment, and day 5 after administration of specific antibody. And in C, what you're seeing is the changes in pericyte in red and the basement membrane that you see in blue, the coverage that happens during vascular normalization, which is almost similar to a normal vessel. And these phenotypic changes are shown in the vasculature, and they reflect changes in the balance of pro and anti-angiogenic factors in the tissue. So as important as angiogenesis is the metabolism of cancer cells, which is elusive. So Otto Heinrich Warburg is synonymous to us with metabolism. The Warburg effect shows that tumor cells, unlike the normal counterpart, utilize glycolysis instead of mitochondrial oxidative phosphorylation for glucose metabolism, even if there is an oxygen-rich condition. Whereas Warburg hypothesis says that cancer was caused by defects in oxidative phosphorylation in mitochondria, forcing the cells to revert to a more primitive form of energy generation, that is glycolysis. In his view, we see that this switch between glycolysis and oxidative phosphorylation caused the cells to become undifferentiated and cancerous. But what was wrong with Warburg's hypothesis? Firstly, mitochondria are not always defective in all the cancers. While some cancers have mitochondrial mutations, like the isocitrate dehydrogenase mutation in glioma, there are many tumors that maintain functional mitochondria and they can actively use OX4s. And the studies have shown that glycolysis is just an adaptive switch rather than a mandatory switch. And glycolysis is not just a backup, it's an advantage for cancer cells. They favor it not because of mitochondrial failure, but because rapid proliferation provides metabolic intermediates for the biosynthesis, like nucleotides, lipids, and amino acids required for them to grow. Also, the lactate production reshapes the TME and helps in immune suppression too. There are tumors that exhibit metabolic plasticity. Many cancers can switch between glycolysis and OX4s depending on their environmental condition. So while hypoxic areas will rely on glycolysis, normoxic areas might rely on OX4s. fatty acid oxidation or glutaminylases for energy. So what is the modern view of cancer metabolism? It is now understood that as a dynamic reprogramming process that balances energy production, biosynthesis, and redox balance. So instead of simply glycolysis or oxphosphate, tumors use multiple pathways, including glutaminylases, one carbon metabolism, and all the metabolism pathways that you can think of. And sometimes oncogenes like MYC or HIF1 alpha drives glycolysis, and tumor suppressors like P53 promotes the mitochondrial respiration and oxidative metabolisms. Also, lactate formation from tumor cells can fuel neighboring immune suppressor cells to enhance immune evasion. Having come so far, our next thought process would be about therapeutic implication. So, targeting glycolysis with hexokinase or LDHA inhibitors was explored, but it was later found that tumors can switch to OXPHOS as a resistant mechanism. OXPHOS inhibitor like IACS 10759 were in clinical trials at MD Anderson, and they used this complex inhibitor, but it showed a very narrow therapeutic index in phase 1 trials for AML and solid tumors, with dose-limiting toxicities of elevated blood lactate and neurotoxicity. Because of that, MD Anderson later discontinued this trial. But later they did reverse translational studies in mice, and that showed peripheral neuropathy, suggesting further research which is needed to understand what is this link that is between the OXPHOS inhibition and neurotoxicity before we can go forward with this complex 1 inhibitors as anti-cancer agents. Also, metabolic targeting is now more personalized, recognizing that not all tumors follow the Warburg effect uniformly. So now it's time for us to move to part 3, which are the recent breakthroughs. But before that, there is another trivia question. Can anybody name this painting or the painter? It is quite a famous painting with a lot of vivid colors.
Yes, anybody would like to answer the question? I think no one is there.
Yeah, sure, Doctor.
Yes.
So this is the screen.
Picasso. Dr. Nandini has said Picasso.
Yeah, unfortunately, this is Edward Munch, and this is the Scream. Why I put is because there's a internal talk inside graduate students that this is actually a depiction of the graduate student life or MDS student life, where you have a existential angst showing us, anxiety and despair. So I just thought of putting it as a funny picture here. So coming back to science. Uh, One of the recent breakthroughs are in targeting KRAS, which was once thought to be undruggable, especially because of the protein, the docking site, the peculiarity of the docking site. And AMG10 or sotorasib was the first drug targeting KRAS to be FDA approved for clinical use, and this was in in non-small cell lung cancer in 2021. Then there is the advent of liquid biopsy, where you can see every biweekly there are new studies, new publications coming out where they are using minimally invasive method for detecting cancer by analyzing either circulating tumor DNA or circulating tumor cells or extracellular vesicles, tumor educated platelets, etc. So what they do here is they use the molecular markers in blood or body fluids to give a real-time genetic snapshot of a tumor without requiring a traditional tissue biopsy. And this enables early cancer detection and continuous monitoring of tumor progression. Another advancement is in the field of tumor organoids, which are three-dimensional models that are derived from the patient's tumor cells and mimic the tumor's key characteristics. These 3D models are very good because they help in personalized drug screening and treatment prediction. And they can also enable rapid screening and validation of multiple drugs. But despite their advantages in studying tumor biology and drug responses, the challenges of limited success rate in establishing organoids and the low hit rate for viable drug candidates is what is holding the field back. We cannot go forward without discussing the immunotherapy revolution in cancer treatment. And this is largely driven by two key approaches, that is a checkpoint inhibitor, including PD-1 and CTLA-4 inhibitors, and CAR T-cell therapy that enhances the immune system's ability to target cancer. Checkpoint inhibitors like Keytruda or pembrolizumab, as you know it, or Opdivo or nivolumab, they block PD-1 or CTLA-4 and restores the T cell function to attack the tumor, which improves outcome that in studies, especially of melanoma and lung cancer, we have seen. Whereas CAR T cell therapy involves engineering a patient's own T cells that are taken out and to express chimeric antigen receptors or CARs that recognizes specific cancer antigens. For example, CD19 in B cell malignancies, and this has led to remarkable success in leukemia and lymphoma. However, CAR T therapy right now faces challenges in solid tumors due to antigen heterogeneity, T cell exhaustion, and potential off tumor toxicity. So they even try to combine checkpoint inhibitors with CAR T cell therapy to enhance the immune response by improving the T cell persistence and tumor infiltration. But the challenges like immune-related toxicity, high cost, and the need for better biomarkers still remain key areas of ongoing research. And for this work in 2018, Nobel Prize was awarded to James Allison and Tasuku Honjo for their discovery of or identifying the CTLA4 and PD1 as immune checkpoint that regulates the T cell activity. A phase 2 trial from BioNTech last year has showed that there was an investigational mRNA immunotherapy, which was in combination with another PD-1 checkpoint inhibitor, and they could show a statistically significant improvement in the overall response rate compared to the anti-PD-L1 relapsed advanced melanoma. Some of the other advancements in the field of immunotherapy are cancer vaccines, oncolytic virus therapy, and cytokine therapy. Just a few sentences about them. Cancer vaccines, they include either tumor or dendritic cell-based synthetic peptide or nucleic acid-based vaccines that can stimulate the immune system to recognize and attack the cancer cells. And recent advancements has highlighted that mRNA-based vaccines as a promising approach than DNA vaccine, even though DNA vaccines are more stable because of their efficient antigen expression and immune activation. Then there is cytokine therapy that utilizes immune modulating proteins like interferon alpha and interleukin 2 that enhances the anti-tumor immunity. And historically, it has shown some successes in treating metastatic RCC and melanoma, but still there are high dose poses toxicity challenges, and they still have a lot of challenges to face. And finally, oncolytic virus therapy that employs genetically engineered or a naturally tumor-tropic virus like herpes-simplex virus or adenovirus, which would selectively infect and kill your cancer cells while stimulating an anti-tumor immune response via MHC1. And these viruses can target tumor-specific markers like PSA or EGFR or HER2, and they can ensure selective replication only in cancer cells. But still, the clinical trials show that potential off-target effects on normal cells need further research. And one of the fastest growing areas in cancer studies is cancer imaging, and the future resides in functional molecular imaging and theranostics. So FTG PET is widely used functional imaging technique. We know it helps in tumor detection, metastasis evaluation, and treatment response monitoring using PERSIST criteria. So here a glucose analog is there with fluorine 18 that is replacing the hydroxyl group at the second position. And what you're seeing here is a case where FTG is trapped in highly metabolic cells like neurons and also in proliferating tumor cells showing the metastasis. And here you can see a patient with EGFR positive metastasis who's been treated with erlotinib, which is a tyrosine kinase inhibitor, which we discussed previously. And this set is one patient who shows partial metabolic responding. You can see how the metastasis has reduced. And this is a case of progressive metabolic disease where the cancer has expanded. Just note that in brain or urinary bladder or kidneys, it is not ideal for detecting metastasis because kidney and urinary bladder has tracer excretion in them, which shows the black or FTG avid areas, which are not cancer deposition. And similarly, brain has high metabolic activity and glucose intake, and that is the reason why you cannot detect brain metastasis with an FTG PET. So modification to FTG PET is fluorothymidine PET or FLT PET that helps in the in vivo assessment of tumor proliferation. And this indicates the activity of thymidine kinase, which is an enzyme that is active in the synthesis phase of cell cycle, which provides more direct measure of tumor cell proliferation. So what makes this FLT PET better than FDG PET is that this can help you understand the true tumor proliferation, distinguishing from treatment-related metabolic changes or tumor progression from inflammation, etc., which can be sometimes confounding in FDG PET. And another advancement that I would like to add here is F18 FMISO PET, which is fluoromisonirazole imaging, and this is to detect tissue hypoxia. So FMISO binds covalently to areas where there are lesser oxygen. So this provides insights to oxygen-deprived tumor areas and can help guide radiotherapy and hypoxia-targeted treatments. So this is an image of FMISO PET in oropharyngeal tumors, and the figure illustrates three scenarios where in A, what you see is tumor that was initially negative for hypoxia that remained so during the treatment, and in B, tumors with pre-treatment hypoxia that resolves with treatment, and C, where tumor with persistent hypoxia before and throughout the treatment, highlighting how good FMISO PET is in monitoring the tumor oxygenation changes and predicting the therapy response in cancer. One last figure from the cancer imaging side, and this is from an advanced MRI technique. You can see a T1 weighted image with gadolinium and a T2 weighted image, which helps to visualize the tumor mass based on the structural abnormalities. But there is something called as fluoroethyl tyrosine or FET PET. This is different from FLT PET. This is FET PET or FET PET. This provides assessment of increased amino acid metabolism, which is just like glucose metabolism, very high and in metabolically active tumor tissues. So in these MRI scan, you can see in T1 and T2, there are tumors of similar size, but in the F18 FET PET, there highlights a significantly larger area of tumor infiltration that has gone undetected in the MRI scans. And this suggests that metabolic imaging can provide a more comprehensive assessment of tumor extent, which is crucial for treatment planning and monitoring disease progression. So coming to the final slide in part 3, that is the future of precision oncology. The applications in omics technology have shown increased potential to drive drug discovery, diagnosis, and treatment decisions. And the characteristic information of the patient can be gained from DNA sequencing, RNA analysis, protein components, or the analysis of DNA modification or epigenomics, cellular metabolism, microbial population in a patient, and even the study of the complex interactions between different organisms and the environment. Even though there are recent trials with targeted therapy that have demonstrated encouraging response rate, the consequence of even more precise patient selection is that a biomarker positive population can be very small, and the most effective targeted therapy may only benefit a very small number of patients with specific tumor types, while targeted therapies are not yet available for majority of the patient. So any novel approaches to drug development and patient testing needs the integration of all these multiomics data along with the preclinical experiments and clinical trials, and there should be a good communication between all disciplines along with global collaboration to enhance it. This is the best way to ensure that as many patients as possible can benefit from from precision-based therapies. So coming to the part 4 of how to get into cancer biology PhD in the US, Dr. Bhuvan, are we running short of time or do we still have time?
Ma'am, no, it's the time is up, but how long will it take? Because
I
again, it
can,
will be an interesting thing.
sure. I mean, I can just run it out very fast.
Yeah, yeah. I think it will be a good maybe if it takes 10 minutes so or so, I think that will be fine.
Sure, sir. I think it'll take lesser than 10
Okay, yeah,
because this is
go
one
ahead.
question that
Because
I keep
many people will
asking
be
them.
interested to know how to get into the PhD in the US.
I
So,
know, sir. And this is a
yes,
question because I get frequently asked.
yes,
I
yes,
thought
yes.
I would put it in.
Could have been one of the questions also. So, please go
Sure,
ahead, then we can take up the question. Please go ahead.
sure.
Okay.
Thank you, sir. Yeah, one more trivia question which we do not have time for, so I would go ahead and answer it. This is image of the creation of Adam by Michelangelo, and this is an image in the Sistine Chapel, and it is just showing the mastery of Michelangelo in the human anatomy, how he has analyzed and drawn it in back those ages. So cancer biology in the US, there are multiple steps that are very much important. Online application, that this is usually 4 to 5 pages. Be very slow and careful when you're applying this. You don't want to put in any wrong information. Statement of purpose and personal statement, these are two documents that every university is asked for, but remember there is stark difference between these two. What I see in most of the students when they apply is they kind of get confused with what is asked out of it. So statement of purpose is focused on your academic and professional goals and how do you plan to contribute to a specific program. So this should answer on why you think you want to pursue this specific program, and you have to highlight all the labs or facilities and projects in that institution which you're specifically applying to, what attracts you or why you want to apply there. Whereas personal statement should show on who you are, or it should tell of your personal experience. So for me, I started off with my story or why I was interested in cancer. So you can go ahead with, I mean, you can think of anything, and the more creative you are, the better it is. And try not to use, the templates that you see in Google Scholar. I mean, Google, you can of course use it to get an idea, but that makes your application very generic because remember, everybody else is going through the same website too. So next is be sure of what they are asking for. Is it a resume or a CV? Because a resume is a very concise summary, mostly 1 or maximum 2 pages, whereas CV is more detailed. You can also find NIH BioSketch in website, which you can use to update your resume. And as long as you continue in your academic journey, you can keep updating it. You will need 3 letters of recommendations, and most preferably if that is from a professor, and well and good if the professor has a PhD, because people generally look into it. Then the transcripts, especially because many of our universities have, the certificates which are written in our mother tongue. So that makes it difficult for them to understand what is written and whether the genuineness of it. So there are some accredited evaluation companies. So, but it's better to go to the specific university that you're applying to and see what they want or how they want, rather than just going it and doing it upfront. Next is be very careful, or don't neglect your IELTS or TOEFL exams, because I've seen people saying, no, I am I'm good with English. Of course you are, but the important thing is IELTS has a pattern, which, if you're very fluent in English, it might take you maximum 2 weeks or even a week just to understand the pattern. Just don't go unprepared thinking, okay, it is just listening, writing, and reading. It is quite a challenging thing. And application fees, mostly it is somewhere between $50 to $100. So start, accumulating and working for having that fees. And GRE is something that most of the universities used to ask pre-COVID. Right now, most of the universities have waived it, and you can check in the individual program policy and take it only if they say that it is desired. And choosing a university. So I would say this is purely my way of, or how I did it. So I went to the NIH website and I looked for the that year when I was applying. So in 2022, which was the universities which had the highest NIH funding, because that would directly translate to the quality of your institute. So this is, I think, 2023. You should be looking out for 2024, which would come out soon. So, and what I did is, at the moment, I just completely discarded the first 5 universities, because I thought I don't want to be that competitive, or I was underconfident to apply to bigger universities, and I really wanted to shortlist into one or maximum 2 universities which I was sure that I would get an interview call for. So I chose between 6 to 20, and then went through each of the universities. Specularities and what are the core facilities that they have and the research they have, and I shortlisted 2. So this is so my university comes here in, I think, 10. And another way is, if you are peculiar that you want to do this research completely in oral or dental related, you can go to NIDCR website and look for the dental schools that with the highest funding, and that is something that you can do. So you can use anything. You can even use a city that you want to live in, Chicago or New York or California. Even you can use whatever you like, but this is just the way that I did it. So some of the do's and don'ts: research extensively, tailor your application to each program. Do not write a generic application everywhere. See what program and faculty aligns with your interest. Demonstrate your research potential in your CV and your SOP. Highlight your research experience, presentations, relevant projects, publications. Plan your finances. I would say check the university websites very clearly. 95% of the public universities here are government funded, and you will have a pretty good stipend. I would say somewhere between 2,500 to 3,500 plus or minus $200 after tax cut, which is a good enough money for you to live in and have savings. And also look into the visa and travel application. And most of the universities would give you free health insurance if you are selected as a PhD candidate. Be mindful of deadlines. Start early. There is a saying here that if you are early, you're on time. If you're on time, you are late. So make sure, if the deadline is December, at least by October to send your application. And proofreading and review is very, very important. I cannot highlight the importance of it. In my case, I had 6 people to thoroughly go through my application, right from my husband, my friend, my cousin, my teacher, a postdoc. So people from different aspects can help you, and especially people who have a very good fluency in English, give it to them also to correct it. Please don't do generic applications, and don't ignore the advisor compatibility. You don't want to go land up in a lab that you don't have any interest in. And do not overload your application with irrelevant details. I see sometimes students, they don't know what to add. So ask your teachers, they will be able to guide you. Add what is relevant that you have done in the field of cancer. It can be publications, or even organizing some events, or anything of that sort. Do not add things that do not add up. Like, for example, if you did so many studies in forensic odontology, I'm not saying it's bad, but those doesn't directly translate to your cancer biology application. So look into it. Next is ignoring cultural and academic differences. So learn about the US academic system. So one mistake that I used to do is whenever I write emails, I say, hi, I'm so and so, and I am a gold medalist, blah, blah. I mean, they, in the US university systems, your academic scores and performances doesn't really match. I'm not saying this to discourage y'all. Please do study well and get good marks, but this doesn't really help in your application. What they look for is, are you somebody who can who can be a good fit, a team player in a lab, who has ideas, who would just open up and talk, and things like that. So sometimes you have to look at that. And another thing that I've seen, especially us Indians do, is we would write 100 sir or 100 madams in the email. Please don't do that. It took me time to unlearn it. And so you can always write dear Dr. Surname. I mean, whatever it is, rather than respected sir. And I mean, these are things that you would learn on the way, but I am just telling you because I went through this recently. And do not neglect to follow up. Some key points I want to look into is, I get asked a lot, like, how many publications do you want? It's not on the quantity, it is quality. I know a friend, a colleague of mine, who has just one publication, and that too, he is not a first author. But the publication was so amazing, and the kind of experiments that he did for the publication, he was able to express it in his statement of purpose. And the reader was able to understand, okay, this dude knows what he's talking about. So give that kind of an impression. Give more importance to quality and where you publish it. Have an ORCID ID already, because that's very important as a person in medicine, and a LinkedIn profile. Keep seeing what is updated, and your, the people who are looking at your application, do look at your LinkedIn profile, honestly. So keep it updated, and understand that there's a difference between LinkedIn and Instagram or any other social media. Try to keep it much professional as possible. And addressing the elephant in the room, use of AI in applications. So, of course, everybody uses AI. We are not denying it. But how to use it, using it wisely and for your own advantage, is the most important thing. So, do not copy paste and just rephrase it. Always read through, use it only when you have so much difficulty in rephrasing or connecting two sentences or knowing what it is. Do not copy and paste, because all the universities have at least one or two AI detection softwares with them, which they use for all our assignments and homeworks, everything. And sometimes AI can even wrongly detect yours, but at least that time your conscience stands for you, and you can defend for yourself, saying, no, I have not used AI in my application. So please take good care of that. And avoid asking basic questions that is already answered on website. People here like replying. They do reply usually in a week or so, but do not ask questions that you already know just for the sake of asking questions. Just I wanted to put that forward. And this is a website I wanted to give it to the MDS students, which would, when you have journal club presentations and when you need recent articles in cancer, you can use this website. And you can use Schimago journal and country ranking if you want to know on the quality of the journal that you want to publish. So you can, if you are publishing in laryngoscope, just put laryngoscope in that, and it will show you, the H index, or the impact factor, the citation, the number of female authors that was. I mean, it gives you a very comprehensive review. And this is the genome browser gateway from University of California. This will help you to align your DNA and protein sequences and give you an entire human genome overview. And this is a website for a comprehensive database that helps you keep the information about protein sequences, the structure of a protein that you're interested in, or the location within the cell. And all those information is available in UniProt. And these are my references. And one advice I would like to give is, start reading. That is the only way to enhance and elevate your minds. And definitely my recommendations are biased. There are certain authors like Siddharth Mukherjee and Dr. Abraham Varghese that I'm interested in, but maybe take a screenshot of this, and I would say it wouldn't disappoint you at the end. And definitely, lastly, acknowledgements. This is something that we all have heard many times. A 1675 letter from Isaac Newton to Robert Hooke, who was his rival at that time. He has said that if I have seen any further, it is by standing on the shoulder of the giants. So I would definitely like to thank my lab, especially my PI and advisor, Dr. Jason Wadham, and Chief Scientist in our lab, Dr. Benjamin Caston, and my HOD sir, Dr. Dinagar Desai. Sir has showed it with his life that no matter where you reach, all that matters is are you being kind and are you being a human, a good human. And my colleague and friend and a good cheerleader for me, Dr. Sitara, and my teachers, Dr. Priyal and Dr. Shubha, for their unwavering support. And my boss and visionary, Dr. Paul Salins, who helped me to look out of the box and developed a likingness for art and history, etc. And of course, my family for who I am and what I am. And thank you. I'm open to any questions, and thank you all for attending this talk and staying back after all this long time. Thank you for your patience, time, energy, and your patient listening. Thank you.
Yeah. Thank you so much, Dr. Sharon. I think we can take the questions first. Dr. Rahul has asked one question. What is the role of tumor-associated macrophages in progression of tumor?
Oh, yes. I mean, tumor-associated macrophages, like I was saying, I mean, the M2 macrophages, they release the different kind of, cytokines and inflammatory factors that can modulate not just angiogenesis, but also in the extracellular matrix remodeling. So there are multiple studies that are coming in, but it is not one significant factor as such, but in each of the core hallmarks, tumor-associated macrophages do play an important role, mainly in angiogenesis and extracellular matrix remodeling.
Okay. Then there are a lot of responses. That fantastic presentation, very nice presentation. All appreciation messages are there. Then one more question is by Dr. Dinakaran is if angiogenesis is playing a key role in cancer therapy, then the process of vessel formation is changed in metabolic disease is altered. How to target cancer in such cases?
So that is where I think Dr. Rakesh, that's a good question though. Dr. Rakesh Jain's vessel normalization theory comes. So what he proposes is you give, for example, bevacizumab in a very low dose, like how we do our oral metronomic chemotherapy. You give a lower dose, and that would help in forming a kind of functional blood vessel, and that indeed would give the drugs and would let or reduce the tumor hypoxia that is associated with this and inhibit the hypoxia induced infiltrating immune cells, etc. So adding this anti-angiogenic therapy in vessel normalization can in fact help in tumors getting targeted by drugs, either targeted therapy or chemotherapy. So it must be mostly a adjuvant or a mixed therapy that would help in case. I know I'm being talking convoluted, but it is basically tumor vessels should be made functional, and then you can give other kind of chemotherapy and immunotherapy drugs which would act on the tumor cells, if that makes sense.
Yes. I request now Dr. Nidhi ma'am to give her closing remarks because actually she is the moderator for today's session, but due to network glitch, she was not able to join that time. So I request madam uh, to ask a question or her comments or remarks on the lecture.
First of all, I would like to congratulate Dr. Sharon. It was just an excellent presentation on such a complicated topic like cancer biology. And the good thing is today we had around 50 plus people attending the journal club today. It was, I think, the highest of the number today. So it's a good thing for Dr. Sherin to, I think, hear about. And also we got an added bonus that how to do PhD and related information that we got. I think it must be really informative for the juniors who want to enter into that field and go to US for this. So it was very thankful to you, ma'am, that you exceptionally thought about this and added a word on this topic. So I'm very thankful. Ma'am, I had just one simple question, like day-to-day basis, clinical basis, like what as an oral pathologist, most of the time we deal with is oral cancer, that is OSCC. So in relation to OSCC, do we have some like a short panel of molecular markers that could be helping us evaluate the treatment prognostic part day-to-day basis, like just a short, sort of small spectrum that we can cover?
Okay. One thing that we, or in my research lab that we do is definitely EGFR positivity. We look for it just to make sure that, the monoclonal antibody therapy that works. But I think your question was more based on the diagnostic side, right, ma'am?
Diagnostic and prognostic sort of also, ma'am. Like
I think P16 is, of course, to HPV positive and to see then the prognosis for oropharyngeal tumors. I think P16 is a good enough marker. Other than that, I cannot think of anything on the top of my head right now, ma'am.
no problem. Well, liquid biopsy is also recently I was also reading few articles related to it that it is said that it is not a very validated technique as of to use. Like so, can you comment on this in any way?
Sure, ma'am. Totally. I agree to what you just said. It is not validated. There are a lot of articles that are coming that is coming through. Even in our lab, we are doing some soluble molecule study also. So the validation part is quite challenging because, it is again, those markers that we are trying to validate for, we are again comparing it with IHC and, those are which are not again standardized. So in that way, I think more papers would have to start coming up and then a systematic review on it, and we will get a better idea after that is what I
It
think.
should be used as an additional thing apart from the routine.
Yes.
Okay.
And also the
Okay,
cost
thank you so
associated
much.
with that is also something that will be, I think, to be considered. Unless it's done on a day-to-day basis. Yeah.
Okay, thank you so much. It was very, pleasantful lecture, a very insightful, deep lecture on cancer biology. On behalf of POPMA, we really thank you for putting on all the efforts and taking such an intensive good lecture today. Thank you so much.
Thank
Over
you,
to
ma'am.
you, Dr. Bhuvan.
Yes. Dr. Sherin, I have a question. Though indeed your your presentation was quite informative, lot of new insights about the pathogenesis mechanisms and all lot of stuff related to cancer. I have a question that now are we targeting extra chromosomal DNA
Hmm. Uh,
therapies and any relevant studies pertaining to oral cancer? And
hmm.
one is like, can we develop, because recently I could see in one of the articles or maybe in the newspaper, it was that personalized cancer vaccines from the Yale
Right.
University, that study has done. So
Right.
can you throw some light on those areas?
Of course, I think cancer vaccine is something, like I said, the mRNA vaccine, that was a trial that was done, I think, in Germany, if I'm not wrong. But
No, it
then,
was Russia. It was
Okay,
Russia.
yeah, that I saw. I mean, that was more than an article. It was a newspaper publication, is how I saw the Russian one. So I do not know the authenticity of that one. I wanted to know more, but even in multiple articles, it just says that it's just an MR. I mean, I don't even know whether it's an mRNA vaccine, but they just said it in few sentences. There wasn't much about the mechanisms behind it. But cancer vaccines, like how we have Gardasil for cervical cancer, I think definitely it is something that needs a lot of clinical validation and trial, but at least for HPV or hepatitis B viruses, most of the viral induced cancers, I think cancer vaccine, it's not too late from us having that in market is what I think, sir.
Because as you know that cancer runs in families, so some
Right.
personalized cancer vaccines, because those who know, because now I'm into the diagnostic industry, and I know people who have got their oncogene panels done. It may cost around 1.5 to 2 lakhs or something, but they have got their panel done, who thinks that they can have cancer in the future. And
Right,
now
right.
nowadays it's actually people with no habits, nothing, they're getting it's such a
Right,
rampant disease
right,
in the
right.
practice. So
Yeah.
that was the thing. And any targeting therapy for extra chromosomal DNA?
So that is a field which I have not gone deep into, sir. Or to be true, that is
Okay.
the first time I'm hearing about extracorporeal DNA targeting, which I'm not aware about. But the previous thing that you were mentioning about that personalized medicine thing, I am part of an organization called Southern Research here, where they are giving personalized medicine, personalized diagnostic test, especially for Black women with BRCA gene mutations. I mean, breast or
Okay.
ovarian cancer history, they are doing it a free of cost, even though I don't think insurance covers it, but they are doing this diagnostic testing for them as of now.
Okay.
Yeah.
No, it was great. So uh, Dr. Sharon,